Welcome to this explainer. Today, we're diving into something genuinely fascinating. We're taking a journey that connects the totally invisible world of microscopic genetic theory right to massive real-world applications. We're going to see how the tiny, microscopic behavior of chromosomes directly dictates everything from the literal food on our plates to the genetic health of our own families.
Think about this for a second. It's wild, isn't it? How can a simple mathematical ratio, something first spotted in a field of pea plants over a century ago, actually give us the power to predict complex human diseases or even engineer the next generation of climate-resistant crops? Well, throughout this explainer, we're going to put on our genetic detective hats to answer exactly that. We'll be working backwards from what we can see to uncover the hidden cellular blueprints that actually control life.
Here is our roadmap for today. We'll kick things off with the genetic detective workflow, move into the protein-phenotype connection, look at designing better crops, calculate human genetic risk, and finally, tackle populations and environmental wildcards. Let's get right into it.
Section 1. The Genetic Detective Workflow.
Okay, so how do we actually solve complex genetic puzzles? It requires a reliable framework. Effective genetic problem solving is completely about working backwards from the visible clues to uncover the hidden culprits. We do this in 3 systematic steps. First, we look at the offspring and we analyze their ratios. Second, based on those numbers, we identify the underlying mechanism of inheritance. And third, once we know that mechanism, we can deduce the unseen genotypes of the parents. Just remember the golden rule here. Parents can only pass down the alleles they actually possess.
Now, if you are a genetic detective, this specific sequence of numbers is literally your smoking gun. 9 to 3 to 3 to 1. When you observe this distinct mathematical pattern in offspring, it instantly tells you that you're looking at the independent assortment of 2 completely separate genes.
And this is a perfect illustration of how we put that workflow into practice. Let's look at dog coat colors, where black is dominant to brown and solid is dominant to spotted. Imagine a cross producing 27 black solid pups, 9 black spotted, 9 brown solid, and 3 brown spotted. Notice the hidden pattern? 27 to 9 to 9 to 3 simplifies perfectly to our smoking gun, 9-3-3-1. This immediately proves independent assortment with complete dominance. So, working backward, we can confidently deduce that both parents must be dihybrids, meaning they're heterozygous for both traits, to produce this exact ratio. The ratios just effortlessly reveal the parents.
Section 2. The protein-phenotype connection. We can't just stop at the math, right? We have to understand the biological why behind these numbers. The molecular foundation of everything we're discussing is mapped out right here. You see, dominant and recessive alleles aren't just abstract letters on a page. They represent actual physical proteins. A dominant allele usually provides the code for a perfectly functional enzyme that successfully converts a substrate into a product, giving you a normal cellular phenotype. A recessive allele, on the flip side, often represents a mutation. It's a misspliced or mutated sequence resulting in a non-functional enzyme. If the enzyme is broken, the product isn't made, and that leads to an altered phenotype or even an enzyme deficiency disease.
Now actually, before we jump into the real-world applications, we need to bust a massive myth. A lot of people assume genetic ratios are like a crystal ball that works perfectly every single time. Educators call this the diploma trap. The fact is, biology is messy. Ratios are just statistical probabilities. For instance, crossing 2 heterozygous plants might yield numbers that mathematically simplify to exactly 3 to 1. But you only see that clean 3 to 1 pattern because you have hundreds of offspring. In a small sample, random chance can make the actual numbers deviate pretty significantly from what you mathematically expect.
Section 3. Designing better crops.
All right, let's scale this whole thing up to our first huge real-world application, agriculture. Billions of dollars are riding on this exact genetic math. Notice how modern plant breeding combines traditional Mendelian approaches with cutting-edge molecular techniques. Breeders don't just want one good trait anymore. They want the whole shebang. They'll take an inbred line with, say, herbicide resistance and cross it with another inbred line that has insect resistance and stress tolerance. By doing this, they create F1 hybrids that express multiple desirable traits all at once.
Let's actually break down the math making this possible. Imagine starting with a pure line of a crop that is basically genetically bulletproof, completely resistant to disease and drought. You cross it with a highly sensitive line. Your first generation of hybrids gets the absolute best of both worlds. 100% of them show both amazing resistances. But the real magic happens when you cross those hybrids together to create the next generation. Because of independent assortment, the traits mix, yielding our famous 9-3-3-1 ratio. The breeders are aggressively looking for the 9 out of 16 plants that show both of those targeted resistances.
But here's the catch, and it's a big one. It is a total myth that you can just pick the best-looking plants from that generation and immediately sell them as a stable new crop variety. Why? Because phenotype does not equal genotype. A plant that looks perfectly resistant might be secretly hiding recessive sensitive alleles. If you plant its seeds, the next generation will segregate and you'll completely lose the trait. The reality is that breeders have to patiently conduct progeny testing over multiple generations to ensure they have a truly stable line that will reliably breed true every single time. So we can engineer these stable, tough crops. But how do these exact same mathematical rules apply to human families sitting in a clinic facing the prospect of an untreatable genetic disease? Section 4. Calculating human genetic risk.
This is where Mendelian genetics gets deeply personal. Genetic counselors use these exact same principles to help families make incredibly difficult, informed decisions. Take an autosomal recessive disease like cystic fibrosis. If two unaffected parents are both hidden carriers, a simple genetic square shows they face a 25% risk for each pregnancy that their child will be affected. But counselors also have to factor in population genetics. You see, the frequency of these hidden carrier alleles varies significantly depending on a couple's ethnic background, which completely changes the overall risk calculations for diverse families.
And the crucial point here is that not all genetic risks are hiding out as recessive traits. Consider Huntington's disease. It's an autosomal dominant neurodegenerative disorder. It has complete penetrance but delayed onset, which means symptoms often don't even appear until middle age. If a woman's father had it, his genotype likely included one dominant mutant allele. That means she has a 50% risk of inheriting that allele, and if she tests positive, each of her children will also face that exact same 50% risk. Counselors aren't just crunching math here. They have to carefully and empathetically navigate the profound psychological impacts of predictive testing for diseases that currently have absolutely no cure.
Section 5. Populations and Environmental Wildcards.
Let's zoom way out now, from the family clinic to entire global populations. At this massive scale, we use something called the Hardy-Weinberg equilibrium as our baseline. It describes a theoretical, perfectly stable population where allele frequencies never ever change. But in the real world, populations are constantly bombarded by evolutionary forces. Selection changes frequencies based on who survives. Mutation introduces brand new alleles. Migration mixes them all up, and genetic drift causes random chaos, especially in small populations. For conservation biologists trying to save endangered species, deviations from our mathematical baseline are exactly what indicate these active evolutionary forces at work. And speaking of the real world, let's talk about the ultimate wild card that messes up all our perfect Mendelian ratios: temperature-sensitive alleles. These are literally genes whose physical expression levels and dominance relationships change depending on the surrounding environmental temperature.
Let's look at how this totally flips our understanding of real-world genetics. Our whole analytical framework up to now kind of assumes a constant environment. But look at the shifts happening here. In a cool environment, a functional allele might be completely dominant over a temperature-sensitive one, giving you red flowers and a perfect 3-to-1 Mendelian ratio. But those exact same heterozygous plants in a warm environment, and the heat might inhibit the normal protein, causing the temperature-sensitive one to take over, turning the flowers blue. The environment literally altered the dominance relationship. If you don't control for environmental effects, your expected ratios will fail entirely, leading to totally incorrect conclusions.
To pull all of this together, there's one profound, big biological rule we need to remember from the source material. Inheritance patterns directly reflect molecular mechanisms, making all genetic predictions ultimately traceable to cellular and molecular processes. Think about it. Every crop yield, every genetic disease risk, every offspring ratio. It's all physically tied to how chromosomes separate during meiosis and how proteins function inside the cell. It's this unbreakable link between microscopic structure and real-world function.
Which brings us to a final, pretty urgent question to ponder. Knowing what we now know about the rules of inheritance, independent assortment, and those crazy environmental wildcards, given that climate change is rapidly creating new, unpredictable environmental stresses, how would you design a modern breeding strategy today to save our agricultural future? How are you going to apply the cellular mechanisms we've explored to solve the massive global challenges of tomorrow? Think on that. Thank you so much for joining this explainer. Keep questioning and keep learning.